amino acids, but it ends up being hydrophobic, and that's thyroid hormone. The other hormones that are made from fat to begin with, obviously, will be hydrophobic. The catecholamines and peptides, they generally will have a surface receptor, meaning they can't get through the lipid membrane, and they're way too big to get through a channel, so they will bind on the surface. It might seem that they would be at a disadvantage doing so, but that's an advantage because they're hooked up to what's called a second messenger system. And that second messenger system can amplify the response. So with one hormone binding on the surface, you may get 10,000 or a million times the response of the cell related to just one initial hormone binding. So all this second messenger system hooked up to protein kinases, there are multiple levels of amplification, and each protein kinase can phosphorylate multiple proteins, so huge response in the cell, and it all happens fairly quickly. The steroid hormones are lipophilic, so they enter through the plasma membrane. They will have a cytosolic receptor that they will bind to, and then that complex will go into the nucleus and turn on DNA transcription. That's a slow process because you got to transcribe, then translate, then post-translational modification, and then you have a protein that can have particular effect on the cell. This is a slow process. Thyroid hormone is, as I said, also lipophilic. Goes through the plasma membrane, through the nuclear membrane, and finds its receptor inside the nucleus. That also turns on the genome, get a slow response. So steroid and thyroid hormone, relatively slow cellular response with respect to the hydrophilic hormones. There are a couple exceptions we'll talk about to the steroid hormones. Steroid hormones that want really fast responses, they will have a plasma membrane receptor as well. They will be hooked up to second messenger systems. So even though they could get into the cell, they bind on the surface so that they can hook up to the second messenger system and have an amplified and also a fast response for the cell. Examples of second messenger systems: inositol triphosphate and diacylglycerol. Those are defined for you in your textbook, and we'll talk about them more when we get to Sherwood. Those are linked to opening calcium channels, the diacylglycerol will stay in the plasma membrane and have plasma membrane effects like opening channels in the plasma membrane. IP3 is hydrophilic, so it can go into the cytosol and it can open calcium channels in the XR. Calcium can also cause calcium release. We'll see that next semester in the GI system and also the heart. Calcium from the extracellular fluid comes in and acts as a second messenger to cause calcium release from inside the cell. And that's referred to as calcium-induced calcium release. Cyclic AMP, we'll have some membrane enzymes that will take ATP, take two phosphates off, create AMP, which then will form a little sickle shape at the end. It doesn't like to be straight molecule anymore. The charges will attract to charges upstream, and you get a little circle at the end. That's cyclic AMP. Same thing happens with guanosine. You get a guanosine monophosphate that sickles. These act as second messenger systems. The overview of the endocrine system is broken down into, let me grab my pens. to central and peripheral endocrine system. So central, think CNS, so hypothalamus, pineal gland, the pituitary. We have two components to the pituitary, one that is actually part of the brain, and that's the posterior pituitary. And then we have another one that's a glandular structure that comes from the roof of the mouth, and that's your anterior pituitary. Those are your central endocrine glands, and then we have peripheral. And that's the rest. So everything outside the head will be peripheral endocrine glands. So thyroid gland, your thymus, parathyroid on the back of the thyroid, your adrenal glands, pancreas, your gonads, those are all peripheral endocrine organs. The hypothalamus is regulatory. It regulates a lot of brainstem function, central nervous system function, but it also regulates the anterior pituitary hormones. It will have regulatory hormones that release anterior pituitary hormones. Hypothalamus actually grows down and forms posterior pituitary. So hypothalamus will form the hormones that will be released by the posterior pituitary, or it will form hormones that will cause the release of more hormones by the anterior pituitary. The hypothalamus, up here it says ADH and oxytocin. Those are specifically from the posterior pituitary. So cells in the hypothalamus produce ADH, produce oxytocin.
You can pull the board over.
I could pull the board over, but then I have to push it back. So anyway, I don't know why the microphone does that. But anyways, so those are produced by the hypothalamus but released from the posterior pituitary. And then these regulatory hormones. We'll talk about specifically what they are shortly. The anterior pituitary got all these acronyms here: ACTH, TSH. PRL, FSH, LH, MSH. So there's a key over here. ACTH, adrenal, it tells you where it's going. It's going to the adrenal gland. It's going specifically to the cortex of the adrenal gland. So adrenal cortex, corticotropic. What is tropic? Tropic means that you have a hormone that's going to release another hormone. So these regulatory hormones from the hypothalamus that release hormones from the anterior pituitary, they're tropic hormones. You also have this adrenocorticotropic hormone that's headed for the adrenal gland, and it's going to cause another hormone to be released, and that hormone is going to be cortisol. So cortical, cortisol, all kind of linked together to help you figure out what the hormone that will ultimately be released by the adrenal glands, cortisol and corticosterone. TSH, thyroid stimulating hormone. So we've got a regulatory hormone from the hypothalamus that causes TSH to be released. And TSH goes to the thyroid and tells the thyroid to make T3s and T4s. T4 is called thyroxine, T3 is triiodinated thyronine. So we have these backbones of the thyroid hormone that then get iodine stuck to them. Could be one, and then it's monoiodinated, diiodinated, triiodinated, and tetraiodinated. Those are released from the thyroid gland, but mono and dis are recycled back into the follicle and wait for more iodine to arrive. If you have adequate iodine in your diet, you are releasing probably mostly T4s. There's plenty of iodine to stick on these molecules. If you're iodine deficient, we'll talk about it later, you might actually get an enlargement of your thyroid gland because keep recycling these the thyroid molecules that don't have enough iodine, and the gland says, well, let's make more of these molecules and more of the binding protein. It's doing everything it can, but it can't get the iodine. There's nothing in your diet to provide the iodide to convert it to iodine. So you get enlargement, and that's called a goiter.
If you have surplus amounts of iodine, do you store T4 then?
Do you store T4? No. We'll talk about the negative feedback. So high T3 and T4 should shut off the releasing hormone from the hypothalamus and shut off TSH. And that should mean that your T3s and T4s go back down. They get used up and their concentration goes down. When they get low, then TRH, which is the releasing hormone, and TSH come back up, and then more T3 and T4 is made. So you will not be storing it anyway. What you end up doing if you have excess T4s and T3s is you become hyperthyroid. And then we'll talk about those symptoms later. Okay, growth hormone, you have to have a regulatory hormone to release growth hormone. Then growth hormone has to go to the liver, and it has to pick up another hormone that will allow it to have its action. So it's going to be acting as a tropic hormone at the liver to pick up its partner, and they have to go together to the tissues to then have growth hormone and allow growth hormone to have its action. Prolactin, mostly talking about it next semester, just briefly this semester, but breast development towards the end of pregnancy and after birth for breastfeeding. Follicle stimulating hormone and luteinizing hormone. These are going to target your gonads, so testes in the male and ovaries in the female. Follicle stimulating hormone in the male turns on sperm production. In the female, egg recruitment. Every monthly cycle recruits some of those eggs that have already been formed and stored in the ovary. LH is luteinizing hormone. Luteinizing hormone is going to drive testosterone production in the male, which then drives spermiogenesis and supports sperm maturation. And in the female, it's going to drive the production of androgens, which then will get converted to estrogens by the cells of the ovarian follicle. And that will drive the development of the ova and get it ready for ovulation. MSH, melanocyte stimulating hormone. We'll talk more about that when we get to the pituitary. Antidiuretic hormone is the ADH that's coming from the posterior pituitary. The Thymus is a gland that is right in front of the heart, very large when you're young, but by the time you're hitting puberty, which may be 11, 12 years of age, it's going to start to shrink in size. The thymus is there to educate your T cells. I always tell my students that if you think my exams are tough, you don't want to ever go to the thymus, because if you fail the exam of the thymus, you're dead. You don't get out of the thymus. Those T cells are killed off. So that's a pretty tough result of not passing. Not that these cells can get out with a D or a C or a B. They got to get out with an A or A+ rating, because they have to go out and determine what cells in your body belong to you and be able to recognize self-antigens and then recognize what doesn't belong in you. So any little failure there, that could be catastrophic to your health. So they can't recognize the self-antigens. They're killed off. They don't get out of the thymus. The hormone there is called thymosin. It helps with this education of the T cells to recognize the self-markers in your body. Adrenal glands, two components, cortex and medulla. Medulla is actually part of the nervous system. It's post-ganglionic, sympathetic nervous system neurons that don't have axons anymore. So they will make the neurotransmitters, they make dopamine, convert it to norepinephrine, convert it to epinephrine. They have the 3 enzymes to get the precursors of dopamine to epinephrine. So mostly what comes out of the adrenal medulla is epinephrine, which can bind to norepinephrine receptors. So it's a backup for the nervous system. The adrenal cortex is divided into 3 layers, and each layer will secrete something different. It will have some that will control blood glucose concentration, another layer will control sodium and with it potassium concentrations, and the other one will back up the gonads with release of androgens. So various things coming out of the adrenal cortex. The pineal gland secretes melatonin, which is involved in your light-dark cycles, your circadian rhythms. It also controls reproductive mechanisms. Parathyroid fights with the calcitonin from the thyroid gland. So outside the follicle that makes T3 and T4, there are these specialized cells called C cells for calcitonin. Those cells will release calcitonin when blood calcium levels are high. And when blood calcium levels are low, the parathyroid gland will release parathyroid hormone. So calcitonin tends to put calcium into your skeleton. PTH will take it out of the skeleton. PTH will also go to the GI system and make you absorb more calcium. Will go to the kidneys and decrease excretion of calcium at the kidneys. Heart, we'll talk about that next semester, just a little bit about it now, that when there is an increase in volume returning to the heart, doesn't mean that your volume overloaded. You might just go there to the wall and stand on your head, and all the blood from your legs come rushing to your heart, and the little atria goes whining and sees all this volume. And when the atria stretches, then it releases this substance called atrionatriuretic peptide. So diuretic gets rid of volume. Natriuretic gets rid of sodium. When you get rid of sodium, then water follows and your volume goes down. You will actually, all you're doing is headstands, you actually become dehydrated from that because you're not really volume overloaded, but the heart is sensing this expanded volume and causing your kidneys to create more urine. You could also sit in a jacuzzi or sit in water up to your neck, and that hydrostatic force will bring more blood to your heart, and you'll find that you have to get out and go to the bathroom quite often because that volume, that hydrostatic pressure increases the volume returning to your heart, ANP is released, kidneys work harder to get rid of what they see as excess volume. The brain also will release a brain natriuretic peptide. Kidneys, we'll talk about that more next semester. They're the source of erythropoietin for red blood cell formation, and also your vitamin D3 has to go to the kidney for one of those OHs added. So calcitriol, when you have 3 OHs on the molecule, that's coming out of your kidneys. Leptin and relaxin and a whole slew of other hormones will be studied next semester, some from the adipose, a whole lot from the digestive system. This semester we'll talk a little bit about insulin and glucagon and a couple other hormones that are coming out of the pancreatic islets. Next semester, mostly the discussion of the gonads, so briefly testosterone and estrogens this semester. And inhibins are the molecules that the gonads release to turn off FSH and LH secretion. So releasing hormones, they are coming from the hypothalamus. They will enter the blood and reach the anterior pituitary. The anterior pituitary is not part of the brain. It meets up with this downgrowth of the hypothalamus that will form the posterior pituitary. So on a later slide, we'll see the embryology of the anterior pituitary. It's coming from the roof of the mouth. It's going to have cells that look more like the pancreas or some other gland than it will look like the brain cells. The brain also will have cells that will migrate from the neural crest, which forms the peripheral nerves, that will migrate into the center of your adrenal gland and form the adrenal medulla. So that is a postganglionic sympathetic nerve cell with really no axon. The epinephrine and norepinephrine that come out of the adrenal medulla come out due to sympathetic nerve stimulation. So preganglionic nerves going to stimulate the adrenal medulla to release epinephrine and norepinephrine. The cells of the hypothalamus in the area just above your optic chiasm, that nucleus will be called a suprachiasmatic nucleus, so above suprachiasmatic optic chiasm where the optic nerve pathways, half of them cross. That nucleus and then nuclei on the sides of the third ventricle, paraventricular nuclei, they form the oxytocin and ADH, and then the neuron axon will transport it down to the posterior pituitary, and then upon appropriate stimulation, the hormone is released from the nerve into the bloodstream in the posterior pituitary. So tropic hormones, releasing hormones, because that is going to release TSH, ACTH, LH, FSH from the anterior pituitary. The inhibiting hormones, not every hormone has an inhibiting hormone that comes from the hypothalamus. A lot of the hormones that are released up here will be turned off by the target organ hormones that are released by the stimulating hormones. So ACTH releasing cortisol. Cortisol in high concentrations will turn off ACTH, will turn off the the corticotropin releasing hormone. Thyroxine, T3, elevated concentrations of those turn off TSH, turn off the thyrotropin releasing hormone. So there's no inhibiting hormone, it's negative feedback that turns them off. A few do have inhibiting hormones, and we'll talk about that when we get to Sherwood. So formation of the pituitary gland around day 26, which is within the first month of gestation, still in that embryonic period. Your brain starts to grow down, and the roof of your mouth invaginates and forms a little pouch of cells that starts to migrate up to meet up with this downgrowth from the hypothalamus. So pituitary is growing down, and this glandular tissue is migrating up, and this is all happening in something really, really tiny. So not that much distance for those cells to go. They meet up and they form the anterior pituitary, which then is a position term. It wraps around the stalk of this descending brain. The end of the stalk is the end of the axons of the paraventricular and supraoptic axon. The other name for the posterior pituitary is pars nervosa. Pars means part. The part of pituitary that's from the nervous system is pars nervosa. The part of the pituitary that is from far away is the pars distalis. So anterior pituitary arising from the roof of the mouth arises far away and migrates up here. The tuberalis is kind of a funny term. The term tuberalis is implying that there's a little tuber, meaning a little potato or a little peanut, those are tubers, in that area. When you pull the brain out of the skull, the stalk of the pituitary often snaps off, and you're left with this little mound of tissue at the hypothalamus. And the old anatomists thought it looked like there was a little buried potato or buried peanut in that area. So they called that the tuber cinereum, the swelling due to a tuber. And this is no peanut, there's no potato, it is a swollen area, it's actually just this downgrowth of the hypothalamus. This area of the anterior pituitary that wraps around that area where your peanut and your potato are buried is called pars tuberalis. Where the anterior and posterior meet up, it forms an intermediate lobe. In humans, not much there but colloid, so amorphous, gelatinous, kind of ground substance with some connective tissue there. Very little hormones produced there, but there is a identifiable intermediate area, and that's called pars intermedia. Pars nervosa, again, oxytocin and vasopressin. Vasopressin is another name for ADH. So ADH stands for antidiuretic hormone, vaso for vessel, and press in, so it causes vasoconstriction. So that one molecule actually has two really important actions. Reabsorb water, so prevent diuresis, anti-diuresis, and then cause vasoconstriction and decrease the size of your vascular system. The ADH is released when blood pressure drops. So you reabsorb water, blood pressure is going to go back up. You decrease the volume of the vascular system, and blood pressure will go back up. So those two are compensatory mechanisms that try to get your blood pressure back up. Keep blood going to your brain so you don't pass out and have something dire happen to you. Infundibulum is another name for the stalk of the pituitary. You look at the posterior pituitary, it looks like brain tissue. It has very little blood supply, as brain has very little blood supply. It has a lot of stripy looking structures. Those are the axons of the paraventricular and supraoptic nuclei axons. The little nuclei that are there, those are support cells for the axons. So these are glial cells that are holding the axons in the right position. So not much cellularly going on in the posterior pituitary. Anterior pituitary, being glandular tissue, is very vascular. You see very large blood vessels here. You also see a lot of different colors, different stains to the cells, different intensity of the staining. So much more dramatic cellular appearance anteriorly. And then the intermediate lobe is primarily connective tissue looking, fibers, and then the amorphous kind of colloid, the more like brown substance. Anterior pituitary produces 7 different peptide hormones. The posterior pituitary releases 2 peptide hormones. So everything from the pituitary is a peptide hormone. Up here in the print here, the infundibulum is growing down to form the posterior pituitary. Stomodium means mouth area that will invaginate and form a pocket of tissue that migrates up. The pocket that's migrating up is still, as far as I know, still referred to as Rathke's pouch. So it's an eponym, but that's the descriptive term for that. Hypothalamus will control directly via a preganglionic neuron that runs from the hypothalamus down the brain stem, and then will synapse with a preganglionic neuron in the spinal cord. That will then go out to the adrenal medulla to release epinephrine and norepinephrine. The hypothalamus will communicate via the regulatory proteins through the blood to the anterior pituitary. The posterior pituitary will have the hormones formed in the hypothalamus but released from the posterior pituitary. So ADH, antidiuretic hormone, oxytocin, the ADH affects the kidneys and the blood vessels. So blood vessels via its vasopressin activity. Kidneys, antidiuresis, causes the kidneys to insert aquaporins and then have water reabsorbed from the filtrate. Oxytocin is active in females at parturition. So it will cause the smooth muscle of the uterus to contract. When the baby turns and starts to descend, its head presses on the cervix, which stretches the smooth muscle there, fires nerves to the hypothalamus. Oxytocin comes out, goes back to the smooth muscle, binds to the smooth muscle, causes the smooth muscle to contract more. Then the baby presses on the cervix more. You get more feedback to your hypothalamus, more oxytocin coming out, more contraction, and this baby is just pounding against that cervix until the cervix dilates enough that the baby can come out. Then that positive feedback is cut. This is one of the very few positive feedback mechanisms in the body. So stretch causes more oxytocin, which causes more contraction, which causes more stretch, which causes more nerve firing, which causes more oxytocin, and this just builds and builds and builds until the baby comes out. In the male, during ejaculation, The oxytocin will cause the vas deferens to move the sperm out, cause the seminal vesicles to move their secretions out, cause the prostate to move its secretions out, and together these smooth muscles cause all the components of semen to be ejaculated. Intermediate lobe in humans produces very, very small amounts of anything. And what it does produce is going to be MSH, a little bit. And then MSH comes out of a huge prohormone that includes 2 other hormones. So when it's chopped up and released, you actually get MSH plus beta lipoprotein and also beta endorphin, all released at the same time, which your book forgot to tell you, so I put a note there. The cells there will pick up blue dye, so that's why they're color-coded blue here. Your gonads, the gonadotropes FSH, LH in the anterior pituitary, they will pick up blue dye. The thyroid stimulating hormone cells, they will also pick up blue dye, as will ACTH secreting cells. So thyrotropes and corticotropes, gonadotropes, and these cells that release these 3 hormones, the MSH, beta, endorphin, and lipoprotein, all will stain. The granules that contain those hormones will pick up blue dyes. The hormones that are released from the cells that release growth hormone and prolactin, these are acidophilic. So they pick up the acid dye and stain red. So there's somatotropes, soma meaning body, muscle, bone, fat tissue. Those all will proliferate under the influence of growth hormone, and then the liver's release of somatomedin. So I told you the growth hormone has to get its partner. So it goes first to the liver to get the somatomedins release, and then the 2 will go to the tissues to have their effect. Another name for somatomedin is insulin-like growth factor. So it's insulin-like. It's a growth factor, abbreviated IGF. And there's type 1, type 2 that I know of. There might be others by now, but the ones that we'll be focusing on, well, actually the one probably we'll be focusing on will be IGF-1, maybe IGF-2 next semester. So the somatomedin or IGF will go with growth hormone. It will permit growth hormone to have its action. That actually is a physiologic term, that somatomedins are permissive. They allow growth hormone to have its effect. Growth hormone cannot have its effect unless somatomedins allow it to have its effect by binding with it in the tissues. Prolactin, specifically for the breast, the enlargement of the breast tissue for milk formation, and then also for milk letdown during suckling. Oxytocin will be involved in the delivery of the milk to the baby too. The baby cries, the smooth muscle contracts in the breast, and actually milk shoots out. And it's kind of embarrassing for mothers if they're out in public and their baby's hungry and starts crying, their shirt just gets massively wet because the milk just comes out. And if the baby is suckling, then the sucking pressure of the baby sucking on the breast, that also will release oxytocin, and then the milk comes out more easily. There actually have been cases where the mom's getting ready to breast pump and the baby starts crying, and so got bare breasts and the milk just shoots across the room. And that's how powerful this oxytocin effect on the smooth muscle is. The milk just really comes out fast. Guys, you got oxytocin too. So next semester I'm going to ask you, you can think about it till next semester. You can stimulate your breast to lactate. So you can breastfeed your babies. So you think about it till next semester. Most of the guys in the back are going, yeah. That's usually the reaction guys have to that. But with the right stimulation with oxytocin and prolactin, you can lactate and also breastfeed. You would give your wife a break. Okay, so pituitary gland, how do the tropic hormones, how do the releasing hormones get to the anterior pituitary? They get there via the blood. Got a capillary bed in the hypothalamus. These capillaries are fenestrated. Anybody know what a fenestra is? Fenestra is a window. Fenestration means your capillaries actually have holes in their walls. Look like little windows to the histologists, so they call them fenestrated capillaries. These holes are large enough that proteins and peptides can get in and out through these holes very efficiently, just based on their concentration gradient. So up here in the hypothalamus, the releasing hormones get through these openings into the capillaries in the hypothalamus, then go through the veins to the capillary bed in anterior pituitary. Releasing hormones come out, stimulate the cells in the anterior pituitary to release their ACTH, TSH, LH, FSH, whatever. That then goes back into the capillaries, and that goes out into the venous circulation, gets distributed throughout the body. This is called the hypophyseal portal system. A portal system is different than peripheral tissue vascular system. In the peripheral tissue, you have arterial, that will go to capillaries, and that will go to veins, and then back to the heart. In a portal system, this first capillary bed can get connected to a second capillary bed, either by veins or more arterioles, or sinuses, which could be venous sinuses or arterial sinuses. So you've got various mechanisms to connect a capillary bed to a second capillary bed. So you got two capillary beds in series, and this final capillary bed goes to the veins that then go back to the heart. So these veins will go back to the heart. So the portal system, you got two capillary beds in series, connected together. In this case, the hypophyseal portal system, it's these portal veins. So there's a venous connection. But in your kidneys, it's arteriolar connection. In your adrenal cortex, it is arteriolar sinuses, so difference. The anterior pituitary hormones go into venous circulation, into dural venous sinuses, which are large venous sinuses that will drain towards your jugular and your vertebral veins. The posterior pituitary has no, oops, sorry. How do I get that? Off? I forgot to turn it on to mute. Sorry about that. So many things to think about. Okay, so durovenous sinuses, and eventually the specific one in the area of the hypothalamus is called the cavernous sinus. Those will drain ultimately into the jugular and your vertebral veins, and then go back to the heart. So I already talked about that. Okay, this is a review table for you. I'm not going to go over it in class because it's too blurry to really look at, but it tells you the part of the pituitary, tells you the hormone that will come, the hormone or hormones that come out, what the target for those hormones are, and then what the effects of the hormones are. So good for your review for exam purposes. The thyroid gland is the first peripheral organ that we're going to be talking about. The thyroid is located in the neck region. It's just below your Adam's apple. It's the reason why that Adam's apple cartilage is called the thyroid cartilage, because it's next to the thyroid gland. The thyroid gland has two lobes. This intermediate lobe here is only there if you have a hyperthyroid condition, so I'm not sure why textbooks always draw it in, but they do, but you won't see that. It will just be a smooth connection across there in normal thyroid glands, no pointed pyramidal lobe there. The central connection between the right and left lobes is called the isthmus. So you kind of have to lift to say that word. Isthmus is a peninsula that connects two points of land together. So anyway, two sides of the thyroid gland are connected by the isthmus. The thyroid gland is kind of lumpy looking, and if you look inside, there are balls of cells. So this is a section through a round ball of cells. The cells normally are cuboidal in their appearance. They pick up pretty much a lot of dye, kind of acidophilic with a light basophilic color to them. They're about square shaped when the thyroid is normally stimulated. The cells have a lot of work to do. They have to take up amino acids to make a binding protein for the colloid to bind the thymine while it's being iodinated, and then fold to stick the two tyrosines together to form the final molecule. And then the cells have to reabsorb all of that and check, do we have 3 or 4 iodines? If we do, we can chop that off the binding protein, release that into the blood. If there's 1 or 2 iodines, recycle that in. The cells also have to make sure there's enough tyrosine in there, in the colloid, for the thyroid hormone formation. They have to get iodide from your diet and convert it to iodine to put onto the tyrosines. So they have a lot of work to do. They've got to form, and then they've got to double check, and then they've got to release all the hormone. Signs that the hormone is being released, you'll see little teeth marks, kind of little bites out of the colloid. These are referred to as resorptive lacunae, so little lakes of reabsorption. And actually what is happening is the cell is taking a chunk of colloid, it's endocytosing it in, and then checking it for the complete iodination, so that thyroxine being formed, or 3 iodines, the triiodothyronine. Chop those off and then exocytose those into the bloodstream outside the follicle. Recycle whatever needs further iodination back into the follicle. If you don't have adequate iodine in your diet, I told you you'll get a goiter. We'll see on a later slide. It's called an endemic goiter. If you don't know it, you should know that there are iodized salts. You go to Safeway and Long's and you look at the salt that's on the shelf, it says iodized and then plain salt. The iodized salt, if you use a little of that in your cooking, then you get adequate iodine in your diet. You can also get it from natural food processors, but food that you consume, but you have to know where to seek out the iodine. So it's easier just to put, when you're making your pasta, boil the pasta in iodized salt water, and then you've got a good source of iodide that then can be converted to iodine to be stuck onto your thyroid hormones. If you don't have iodide, you're recycling mono and diiodinated thyrine, and the cells are making more binding protein and uptaking more amino acids and converting, they're busy reabsorbing and checking and resecreting. They hypertrophy because they're working harder, but the colloid is going to build up inside the follicle and end up squishing the cells so they look like they're squamous. So in an endemic goiter, the enlargement is primarily due to colloid buildup. When you biopsy the thyroid, you'll see a lot of colloid and squamous cells, squamous appearing cells. If you have a hyperthyroid condition, this colloid will be minimal if it's being reabsorbed at a high rate. The cells also are working hard to form the thyroid and also endocytose it and release it. So the colloid will be minimal. The cells will be very tall and columnar in appearance. So it's only when everything is working just fine that the follicular cells actually look cuboidal in appearance. C cells are extra follicular cells, so they're outside the follicle. They're larger cells, they pick up less stain, they have more cytoplasm, and they won't be as pink staining as the follicular cells. Those are the ones that release calcitonin. Where does your thyroid come from? Grab a paper towel, go to the mirror, grab the towel, pull it out, look at the back of your tongue. There's an indentation in the back of your tongue. That's where the thyroid gland formed and invaginated, and then went down over the hyoid bone, and then down past your thyroid cartilage to come to rest in the front of your neck. So it started in a pair of second pharyngeal pouches, and then formed this thyroid diverticulum, which is this invagination, and then formed what's called a thyroglossal duct, which is just the cells migrating down to their final destination in the front of the neck. These C cells, they form from the fifth and fourth pharyngeal pouches, which fuse together to form specialized cells called the ultimobranchial cells. Those migrate down with the second pharyngeal pouch cells. Also, the nervous system contributes neural crest cells to these C cell populations. So the population is not strictly one type of cell, but a combination of neural crests and these ultimobranchial cells. thyroid gland will be responding to thyroid stimulating hormone. When TSH binds to the follicular cells, they make more T3 and T4. Where does TSH come from? Anterior pituitary. What releases it? TRH. So thyrotropin releasing hormone. So thyroid, meaning thyroid, tropin, it's a regulatory protein, it's going to release TSH. So thyrotropin releasing hormone, so RH. Will then stimulate the release of TSH, and TSH, also a tropic hormone, will cause the release of the thyroid hormones. The T3 is your metabolically active form, but tissues are just half as happy with T4s because they have an enzyme that will chop off an iodine and make T3s. So T3 and T4 can be released. Tissues getting T3 can use it immediately. T4, they have to chop off an iodine before they can use it. To turn off TRH, you have to have adequate T3 and T4 in the circulation. So as T3 and T4 levels rise, then TRH release starts to decrease. TRH decreasing means TSH decreases. TSH decreases, then less stimulation of the thyroid gland. So this is how you regulate the normal range of your thyroid hormone, is by negative feedback to the hypothalamus and also to the anterior pituitary. Okay, take a break and then we'll talk about goiters.